So pigments which help in photosynthesis, there is a wide range, but the main emphasis is on chlorophyll because reaction of photosynthesis starts with chlorophyll. So let us talk about all the pigments. Pigments are molecules which absorb light or any form of energy. One such pigment which is the most important is chlorophyll. How many types of chlorophyll? You would find two numbers written. Somewhere it is written as 10 types of chlorophylls are there, some places 7 are written. We will see why this difference is there. Chlorophyll A, B, C, D and E. So there are 5 types of simple chlorophyll, chlorophyll A, B, C, D and E. This is one. Second is bacteriochlorophyll. Bacteriochlorophyll is, as the name suggests, it is found in blue-green algae which are capable of performing photosynthesis. You can take this bacteriochlorophyll as one, and scientists have also identified 4 types. So bacteriochlorophylls are also of 4 types. And next is bacterioviridin. So if we count the number as chlorophyll 1, 2, 3, 4 and 5 here, 6 is here and 7. That is why at some places 7 chlorophyll types are written. But if you take the subtypes of bacteriochlorophyll, the number will be 1, 2, 3, 4, 5, 6, 7, 8, 9 and 10. So either you will find the number written as 10 types of chlorophyll or 7 types. The difference is only this. If you take it as 1, the number will remain as 7, and if we take the subtypes, we can say there are 10 types of chlorophylls. The most important are these 2. All others are accessory pigments. Accessory means helping pigments. We will talk about this in a minute. Let us also write down about the other pigments which are there. Another group of pigments is known as carotenoids, which includes 2, carotene and xanthophyll. Carotenes are orangish red, the carrot color basically, that is what is the color of this pigment. So it is orange red. It's a combination of 2 colors. Its formula is C40H56. The difference between carotene and xanthophyll is just of oxygen. Xanthophylls are yellowish brown or yellow brown, and their formula is C40H56O2. So these are oxidized forms. So carotene and xanthophyll together they make carotenoids. The most important carotene that we talk of is beta carotene. Beta carotene gets hydrolyzed to give rise to vitamin A. So it is also known as precursor of vitamin A. Precursor is some raw material which will produce that particular substance, and beta carotene is found in carrots, and that is why carrots are considered good for eyesight, because eyesight is dependent on vitamin A. Xanthophyll, yellow brown in color. One example is lutein. This is an important xanthophyll which is found. So beta carotene is one important carotene, and lutein is one example of xanthophyll. Third category of pigments are known as phycobilins. Phycobilins are again of 2 categories, phycoerythrin and phycocyanin. Phycoerythrins are red colored pigments and phycocyanin are bluish purple pigments, or they are sometimes also written as blue pigments, but they are bluish purple, blue purple. Now, so many pigments are there, but the plants look green. The reason is the percentage of green pigment chlorophyll is maximum. Other pigments are also there, but their number is very, very less, and that is why predominantly green pigment gives that green color to the leaves. Let us talk about this chlorophyll now. Chlorophyll molecule is a complex structure. It has two parts, a head part and a tail part. The head part has magnesium ion in the middle, in the core of it, and attached to this magnesium ion are 4 rings. Each ring has 1 nitrogen. So, magnesium ion is connected to the nitrogen of these 4 rings. These rings are known as pyrrole rings. So, there are 4 pyrrole rings. They are numbered as 1, 2, 3 and 4. So, there are 4 pyrrole rings. Each ring has 1 nitrogen, and to that nitrogen is attached the magnesium ion which is in the core of it. In the ring 2, there is a position which is called X. We will come to that X position in a minute. This structure makes the head. It is called porphyrin head. Porphyrin head. And as this head is made up of 4 pyrrole rings, it is also known as tetrapyloric. The reason is 4 pyrrole rings make the head. The head is called porphyrin head or tetrapyloric. Numbering is important, 1, 2, 3, 4. Attached to the 4th ring is a long chain of hydrocarbons. Normally, there are 20 carbons, alcohol containing carbons. So, this complete structure is known as the tail. So, there is porphyrin head and a tail. The tail is known as phytol tail or phytol chain. So, there are 2 parts, head part and a tail part. 4 rings make the head. Each individual ring is a pentamerous ring. It has 1 nitrogen. Each ring has 1 nitrogen which is connected or which is joined to magnesium. In ring 2, there is a position X. If the position X has methyl group, if it has methyl group, then that chlorophyll is chlorophyll A. If position X has aldehyde group, then that chlorophyll is chlorophyll B. So, the basic difference between A and B is only one, rest complete structure is same, and that difference is position X difference. What is at X position? If it is a methyl group, it is chlorophyll A. If it is an aldehyde group, then it is chlorophyll B. Chlorophyll A and B also have some more differences, like chlorophyll A in its natural color. Let us make that difference in the column form so that it is better. Let us write it as chlorophyll A and chlorophyll B. What is in chlorophyll A? Chlorophyll A at X position has methyl group and chlorophyll B at position X has aldehyde group. This is one difference. The second difference is chlorophyll A in its pure form. That means if we extract chlorophyll, only chlorophyll A, then it is in its pure form, it is blue-black. It is blue-black in color. What are we talking about? We are talking about chlorophyll A completely extracted from the plant, only pure form. Whereas chlorophyll B is green-black. It is also called olive green. It is little green with a blackish tinge in it, and here it is bluish tinge. So this is the pure form. Chlorophyll A looks red in reflected light, red in reflected light, and it looks bright green in transmitted light, green in transmitted light. So pure color, pure extracted form has a different color. In reflected light it looks different, and in transmitted light it looks different. What about this chlorophyll B now? It is green-black in its pure form. In reflected light it looks brownish with a little reddish tinge in it. Brownish in reflected light and yellowish green in transmitted light. In transmitted light. So these are the differences between chlorophyll A and B, and we are talking about these two because they are the main pigments. All other pigments, they are going to help in the process of photosynthesis. How they will help, that also we'll see in some time. So chlorophyll structure is one head and a tail. The head is porphyrin head, which has magnesium in the center. This magnesium is connected to nitrogen of the 4 pyrrole rings, and that is why head is also known as tetrapyroring. The 4th ring is connected to a 20 carbon hydrocarbon chain, which is an alcohol containing hydrocarbon chain, which makes the tail or phytol tail. So this is the structure. If position X has methyl group, it will be chlorophyll A. If it has aldehyde group, it will be chlorophyll B. Other pigments are helping pigments. So we have seen the pigments which help in photosynthesis. Now let us talk about the light. Which type of light is important? So light as the next factor. The next condition which is required for photosynthesis is light. Solar energy or sunlight. The solar energy which comes on the earth has various wavelengths. The main wavelength which helps in photosynthesis is known as photosynthetically active radiations, PAR. So So when we talk of light energy, we are actually talking about this photosynthetically active, and this is in the visible spectrum. So we are basically talking about the yellow light, which comprises of the 7 colors. This wavelength, it extends from about 300 nanometers to 700 nanometers. So this is the wavelength which helps in photosynthesis. Scientists performed experiments to find out which wavelength is important. One scientist, Engelmann, he performed a very simple experiment to see which wavelength is the most important or most effective wavelength. What he did was, he took a long trough, a long transparent container, filled it with water and kept unicellular algae in this container. Unicellular alga, like chlorella type of algae, was used, and this entire thing, it was taken as a long structure and it was exposed to a different wavelength. So the wavelength which were used were of the visible spectrum, so this part was exposed to violet, indigo, blue, green, yellow, orange and red. So only that much band was given, that particular wavelength. These algae which are used or which were used here, like the chlorella type, they are photosynthetic. That means they perform photosynthesis. Now to see photosynthesis happening is not possible unless and until we see inside the cell or we extract the substances, intermediate compounds which are formed. So the easiest way to find out that something was happening was to see the bubbles of oxygen being given out. If more photosynthesis is taking place in some area, more oxygen will be given out as byproduct, and those oxygens molecules will come out in the form of bubbles. So Engelmann found that there were 2 bands or 2 areas where maximum bubbles were seen. One was between this indigo blue. So this was the area where maximum bubbles were seen. So this is the place where maximum oxygen bubbles were released. And the second was near the red one, towards the end of orange and red one. So this was the next one. What was happening in these areas? In this area, oxygen was being released. Why oxygen is being released or coming out? Because it is a byproduct of a process. That process is photosynthesis. And if it is happening in 2 regions predominantly, that means that particular wavelength is the most effective wavelength. A slightly different modification was also seen. That was same thing but slightly different experiment where a filamentous green alga was taken. Filamentous alga like Spirogyra, and this was all water. So filamentous alga was taken and the bacteria which were kept in this water were aerobic bacteria. So here all those bacteria which were there, this was the filamentous alga, and these were oxygen using bacteria. Basically these were aerobic bacteria. Again the same thing, this filamentous alga was exposed to different wavelengths. This green filamentous alga started performing photosynthesis, in whichever wavelength photosynthesis is maximum, that is the place where the scientists must have seen more oxygen being given out. Now, these bacteria which need oxygen for their survival, where would they move? They would move to an area where oxygen is available. So, again, in the area of this blue, indigo blue, and in the area of green, all these aerobic bacteria collected. So, the reason for their accumulation in a particular region was that here they were getting oxygen, and how this oxygen was available to them? Because of this filamentous alga, green alga, which was performing photosynthesis. So, both these experiments prove that there are two distinct bands where action is more. Action is this photosynthesis. So, now these scientists plotted a graph. The graph was plotted on absorption by pigments and the wavelength. And the wavelengths were taken from 300 to 700 nanometers. They found that for these pigments, the action was maximum at two regions. It was in this blue band, then again there was a lower action, and again it went up in the 700 region, that is again red. This was the graph which they got for chlorophyll A. This graph was indicating the action. Action is the reaction which is taking place here. So, they called it action spectrum. This was for chlorophyll A. Then they used chlorophyll B and again did the same thing. The graph was pretty much same with a slight modification. So, this purple is for chlorophyll B. Both are overlapping. That means in the blue region and in the red region, maximum action or reaction of photosynthesis took place. They plotted a same graph for absorption. They extracted the pigment, and in the spectrophotometer, when you check which wavelength is getting absorbed, they found the absorption spectrum. And when they superimpose that absorption spectrum here, absorption was also in the same wavelength and action was also in this same wavelength. What does it indicate? That this wavelength gets absorbed more, and this is the wavelength which is actually responsible for the reaction, that is photosynthesis. So, action spectrum and absorption spectrum. Are going to be same. Reason, the place that particular wavelength is absorbed, that is the same area where maximum photosynthesis is reported. So, action and absorption spectrum can be overlapped. They are exactly same in terms of wavelength. Maximum photosynthesis takes place in the blue zone and in red zone. Now, when we are talking of light and absorption, we also need to know how these pigments are placed and how do they absorb this light. We talked about PS1 and PS2, photosystem 1 and photosystem 2. So, now we will talk of photosystems. Photosystems are those collection of pigments which absorb the sunlight, and that is the place from where the actual reaction of photosynthesis starts. So, let us talk about these photosystems now.